Hi all, and welcome to Stem Cells Part 2. So we've been talking about cells that have plasticity, that are not fully differentiated, and so can go down different developmental pathways when given the proper cues to do so in the body, right? These adult stem cells, or we'll be turning to embryonic stem cells in just a moment. But scientists wanted to know, can you turn basically the clock back on a fully differentiated cell, a cell that no longer has any plasticity, that it is what it is? Is that cell with its genetic material capable of driving the development of a whole new organism under proper circumstances? So investigators turned towards cloning, and in 1996 in Scotland, Doctors Wilmot and Campbell and their colleagues actually developed a technique to allow a terminally differentiated cell to drive development of a whole new organism. And a little lamb named Dolly, the sheep, was born. Lots of controversy surrounded this, but let me discuss what they did. So they had a fully mature donor, this 6-year-old ewe here, and they took a mammary cell from her udders, and they treated it in the lab to basically try to turn the clock back on the nucleus. So we know that what differentiates one cell from another is differences in gene expression. And we know that while the chromosomes of a stomach cell and a skin cell may have all of the same genes, the chromosomes don't necessarily look the same, right? Certain areas are heterochromatized in one cell and they're euchromatin in the other, right? There's differential gene expression, which means that the chromosomes look different in terms of what's wrapped up tightly and what is accessible for expression. So they basically deprived these mature mammary cells of nutrients. And you see here, it says that they semi-starved them right there. So they semi-starved them to try to, I don't know, make the nucleus uncomfortable, change the architecture. They didn't really know exactly what would happen, but to try to change basically or remodel the chromatin, and cause the cell to arrest itself, like not be in the cell cycle, be a little bit uncomfortable, remodel the chromatin, and de-differentiate, as you can see, take off its hat and glasses and sort of rest in a state that was not the state that they were in before. So this was going to be the source of the genetic material, this fully differentiated nucleus from a mammary cell. And they took an egg cell from another donor, and you can see here, here's the egg cell from the ovary, and they removed the nucleus from that egg cell. So now that egg cell no longer has a nucleus, and that what they did was they took, but all of the components that are in an egg that will help to drive the early stages of development were still there. Right, RNA, proteins necessary, like transcription factors for things that can help to drive gene expression off of the nucleus, those would have been found in the egg. And so you didn't remove all the contents of the egg, you removed the genome of that particular egg. And then they fused these two together, this enucleated egg, an egg without a nucleus, together with this mammary cell that had been deprived of nutrients and so was in a different state that it had been in before, as you can see here. So they use the cells right here. And then they grew it in culture for a bit, and what they've got was some early embryos. And so they took these embryos, and I believe there were 13 implantations into 13 different surrogate mothers. And the surrogate mothers they chose had the black face, as you can see here, just like this one, different from the white-faced sheep who was the donor of the mammary cell. And normal gestation time later, the surrogate mother gave birth to Dolly the lamb, the little lamb. And Dolly the little lamb was genetically identical to her 6-year-old nucleus donor. So she was a clone of the white-faced sheep who donated the mammary gland, the mammary cell, right, from the mammary gland. So this was done, it was difficult to do, very little efficiency. So only one was born, as I said, even though 13 surrogate mothers were implanted with these early embryos. But it was a tour de force because it showed that the nucleus of a fully differentiated cell could drive development of a whole new organism that was genetically identical, right, to the animal from which the cell was taken. So it showed that there were not changes that couldn't be undone in a terminally differentiated cell. Basically, you could remodel the DNA to allow access to the genes that had been turned off at a later date. The problem with poor little Dolly the lamb, sheep normally live, well, years, 13 years, something like that. She had to be euthanized at 6 years old, Dolly the lamb, because she was found to have progressive lung disease, and she also had arthritis. So the belief was that while this nucleus was able to direct development of a whole new individual, Dolly, it wasn't fully back to a state that it needed to be in order to allow this lamb to have a full life. Now, this lamb, by the way, was able to get pregnant herself and give birth to, I think, something like 4 sheep. So she was it appeared to be a perfectly normal little lamb until she developed these diseases that normally come up in older sheep. But again, a super huge tour de force to show that a terminally differentiated adult cell could be remodeled and reprogrammed such that the genes could be accessed, and so we could call it pluripotent, right? So it acted just like the nucleus that would come together basically from the fusion of an egg and a sperm, at least in being able to direct development and producing a viable organism. Ian Wilmot actually came to Hunter back in the day, was like a rock star being in the building, somebody who was capable of doing that type of, cloning, showing up in room 714. I mean, it was packed. Everybody went. Okay, so which mammals have been cloned since that cloning in 1996? Cattle, cats, dogs, deer, horses, many have been cloned for reproductive purposes most of the time. Your book mentions the cat, the first cat that was cloned, who was called CC for carbon copy, which I think is adorable, but I think it should have been called Copycat. You can decide, that would have been a cute name too. So this is overall just this is exactly a little summary of what we saw on the sheet before. So let me just show it to you. Oh. Right, so here's a pig. They're showing you a pig. And here's an adult female. And so they take an egg that hasn't been fertilized, and then they remove the nucleus, as you can see here, sucking it up into a pipette. So you have all of the goodies that the egg would have to work on the nucleus, right, to allow for division and such. And then they take, in this case, they're showing you skin cells from an adult pig, and you take an electric pulse, right, that can fuse the adult skin cell, this fully differentiated cell, together with the enucleated egg. There you go, fusion, and then you get cell division, and then you get to the stage known as a blastocyst, which I shall show you more of in a moment, and then that gets into a surrogate mother. The surrogate mother can give birth. She gives birth to a cloned animal that is genetically identical to, as you can show from the colors, right, to the one who donated the terminally differentiated cell, like a skin cell. So it is pretty fabulous type of experimentation that can be done now. Okay. Okay, so now what about embryonic stem cells and their power as pluripotent cells, cells that can be maintained in culture, at least human cells, since 1998, and indefinitely in human culture, treated in a way that we know in the body. Of course, embryonic stem cells are not maintained indefinitely. These are going to go down the pathway to create the cells that become part of the developing organism and ultimately into the adult. So how do we get embryonic stem cells, and how are they put into culture? Well, that blastocyst stage that I just showed you from the slide before is the stage that has a series of cells. here that are known as the inner cell mass. And these are the source, inner cell mass. These are the source of the embryonic stem cells. That's where the embryonic stem cells are. The cells that surround are extra embryonic structure. So the cells that you see here, of course, this is a cross section, right? But the cells that are surrounding there, these are going to help to give rise to the tissues of the placenta, for instance. So taking these stem cells here and putting them into a dish, scientists now know how to culture these and keep these in the lab indefinitely, and how we can take them down different developmental pathways. Right, here's your cells of the inner cell mass right here. You can see them right here. Right, and cultured in stem cells, as I said, 1998 in humans. It was done in 1981 in mice. So that's a lot. That's a big gap between being able to develop the technology to sustain embryonic stem cells in culture in mice, and the same in humans, right? It shows you that humans and mice are not exactly the same. I mean, I think a lot of us knew that already, but just because you can culture a cell from one species doesn't mean you can culture it from another species. They require different conditions, and those conditions had to be worked out. All right, but in 1998, they were worked out for humans, and the real potential became known. But this slide is actually showing you mouse ES cells. So taking these cultured cells, they don't just spontaneously become a different, all the different types of tissue. No, you have to apply certain signaling molecules, right, to take them down a different pathway, right? So if you look at just retinoic acid, for instance, if you look right here, taking ES cells that are in culture and applying retinoic acid takes them down the developmental pathway where they become neurons, right? If you put in right here, macrophage colony stimulating factor, this cytokine here, and then you follow it up with interleukin 3 and interleukin 1, then you can take an ES cell down the path to becoming a macrophage, right? So the conditions for all the different types of tissues have been worked out, and what types of signaling molecules have to be applied at what time in order to drive the development, right? So scientists are actually saying, aha, it's ES cell, I want a cardiac muscle cell. So they know how to drive the ES cell down the pathway to becoming a cardiac muscle cell. So that is absolutely amazing. There's controversy to using embryonic stem cells, especially human embryonic stem cells, and that's when you take the cells of the inner cell mass away from the blastocyst, you are this early embryo, you're killing the blastocyst. So you destroy an embryo for research. And so this is highly politically charged in this country and always has been. And if you want funding from the government, from the NIH, for instance, National Institute of Health, which support tremendous amounts of research, you have to select human embryonic stem cells that are already in a registry that have been approved basically by the government for use. And those are the only ones that you can work on, ES cell lines that you can work on if you want to have funding from the government, from the NIH. So there's somewhere around 500 stem cell lines or so that are approved at this point, give or take, that are approved for usage, and you have to select from there. And what's cool about that registry is that they not only have embryonic stem cells that come without verified diseases, but they have embryonic stem cells from lines from embryos that had known defects. And by the way, where did these ES, where did the whole blastocysts come from that donated, the ES cells originally? And they mostly came from couples who were undergoing fertility treatment. And so they had a series of blastocysts made, and let's say they didn't implant them all, maybe because they had successful births from the few that they implant. Physicians, when they're trying to get a woman ready for IVF, in vitro fertilization, another Nobel Prize winning research, by the way, they try to make as many blastocysts basically as they can, so that there are choices to put them in the woman if the first series of implantations don't work. So they stimulate a woman to make a whole bunch of eggs. by manipulating the reproductive hormones, and then they extract those eggs, and then they can put them together in vitro with sperm from males, and then they try to make as many blastocysts as they can, and then they will freeze the blastocyst. Well, they actually freeze it at an earlier stage than a blastocyst, usually, but they try to make as many as they can and implant some, and if the female doesn't want to have it use those anymore, she can donate them with informed consent. So nobody gets paid to, produce blastocysts that get used for research. It's just if these are to be discarded anyway, many people will just say, no, you can use those for research, and allow them to be basically destroyed because the inner cell mass cells are taken out and used for research. But there's a lot of potential for usage of these cells in therapy, and this is because these inner cell mass cells are pluripotent, we've been discussing, which means that they can become any tissue. So what we just looked at was coaxing cells down a differentiation pathway in the lab, right? And being able to show that, from this ES cell, you can make a fat cell. From this ES cell, you can make a macrophage. But what about if you inject these cells that have been taken from a blastocyst and cultured? Can you put them into another blastocyst and see if they make the tissues in vivo, right? And so they can do that. They can take these cells and inject them, and we're back in mice now, because this is not being done in human beings. So you take those cells that originated from blastocyst A, let's say, and then you inject them into blastocyst B, a different genetic blastocyst. And what they've noticed is that the cells can be incorporated into all of the different tissue of the new, what we refer to as a chimeric mouse, a mouse that doesn't exist in nature, but actually has been created in the lab. So these ES cells that were taken from a blastocyst are pluripotent. They can be any of the approximately 220 different cell types that we find in the body. So that's why they're trying to show you with those little red patches, trying to show you that this chimeric mouse has some of these ES cells that were injected into the cell blastocyst in every single part of their body. They can make all three germ layers. So very powerful cells extracted from one blastocyst, injected in another, and they just incorporate and become all the tissues. And then they follow the cues of the different areas of the body to become all the different parts of the body. So very powerful cells. But again, very controversial to use these human ES cells. So enter in Dr. Yamanaka. This is actually, this is my favorite male scientist. I have a favorite male and a favorite female. This is my favorite male. Very, very humble man, just incredibly brilliant and just lovely, full of humor and such. And he said he liked to work on skin cells and not hair cells because he was losing his hair. Called himself a failed surgeon. He was both a surgeon and a researcher. I don't think he's failed at anything, but anyway, lovely, lovely man when you hear him speak. So he decided, let me go around this controversy of destroying blastocysts to get at these powerful ES cells that are found here in this inner cell mass. Let me see if I can take a terminally differentiated cell, like a fibroblast, a skin cell, and turn the clock back on the nucleus. So you're not taking this cell or this cell's nucleus and putting it in an enucleated egg or fusing it with an enucleate. No, you're taking this cell and you're trying to itself convert that cell back in time to an embryonic stem cell-like cell, one that has the characteristics of embryonic stem cells, like so being able to proliferate indefinitely in culture, and then being able to be coaxed down the different developmental pathways in the lab, and then hopefully injecting those cells into a blastocyst and seeing if they can form all the three germ layers, and so can be any of the cells of the body in a live animal. So he first did this in mice in 2006. And in human beings not in human beings, but with human cells, human skin cells in 2007. Won the Nobel Prize for this, 2012. So let me describe it to you. So what he did was he took these skin cells and he found 4 very important factors. They actually happen to be transcription factors so that they are in control of deriving expression of genes, right? And the insertion of these four factors, right? The genes for these four different transcription factors into skin cells actually enable the skin cells to turn their clock back and become what he referred to as induced pluripotent stem cells, so IPS cells. And how he got the genes in there was he delivered them with a vector. He delivered them with a retrovirus. So whole bunch of retroviruses carrying these four different genes that are hierarchically really important in terms of gene expression put into the skin cell. And those four genes were enough to reprogram the nucleus of this terminally differentiated skin cell, turn the clock back on the nucleus, have the chromatin remodel itself enough to turn on genes of the embryonic stem cell. So were they fully like embryonic stem cells? No, they're not exactly. Induced pluripotent stem cells are not exactly like ES cells. They have, if you look at global gene expression, they don't have the exact genes on and the exact genes off, but they're a lot like embryonic stem cells in that they have a lot of the same genes on and off. They can, in culture, reproduce indefinitely, and when injected into mice, if you're looking at mice cells, they can become all of the tissues of the body in the mouse. So something that used to be a skin cell all of a sudden is acting like an embryonic stem cell, and the only things necessary to get it to do that, to change the program, was insertion of four different genes, the gene OCT4, SOX2, c-MYC, and KLF4. So it's astounding that that was all that it took to remodel the chromatin enough to be able to turn the clock back and have these cells become pluripotent, right? Become able to be coaxed down all the different pathways. Okay, I'll stop here and return for the next part.